right, very good afternoon. Welcome to this Tech Talk. Welcome to all of you who are physically in this seminar room, as well as the participants online. Tech Talk is a series of forums and dialogues given by engineering researchers of diverse academic backgrounds to share the insights on innovation-related topics. And today we are very honored to have a distinguished speaker with us, Professor Tong ZHANG. Professor Tong is a chair professor in the Department of Civil Engineering of the University of Hong Kong. And Professor Tong is particularly in the area of environmental microbiome engineering and biotechnology laboratory of the Department of the University. He is also a professor by courtesy in School of Public Health of University of Hong Kong. His researches include wastewater-based epidemiology, antibiotic and resistance genes, environmental microbiome engineering and biotechnology, anaerobic and digestion and bioenergy from wastewater and biological wastewater treatment. He has been listed as a highly cited researcher by Clarivate since the year 2018 in a number of areas, fundamentally in the wastewater and environmental engineering. So shall we put our hands together? So Professor Zhang, over to you.
Okay, can you hear me? Yeah. Good afternoon, everybody. First of all, I would like to thank the HKAE organize this seminar and give me this opportunity to share with you our research. And also I will thank our department head for the kind introduction. And before the seminar, our department head said he doesn't understand what I'm doing. I think that is a dangerous signal to me, okay? So I should try to use this the following 40 minutes, try to let our department head and also you know what we are doing. As you can see, today I will talk about environmental analytical microbiology and its application in our engineering field. My lab is Environmental Microbiome Engineering and Biotechnology Laboratory. This is just a new name. When I was a PhD in this lab, at that time, the name of this lab is Biological Wastewater Treatment. And then we changed it to Environmental Biotechnology. About 10 years ago, we changed to the current name. And although the name is different now, but the focus is the same. It's about the sewage and wastewater. And for you, I think most of you may know that the sewage is a flow of waste. Yes, it is a flow of waste as a result of urbanization in the past 200 years, started in UK. And also, more and more, people look at the sewage or wastewater and recognize that wastewater is also water resources. I give you a quick quiz. How much water content in the sewage or wastewater in percentage-wise? 99% or 99.9% or 99.99%? Which one is correct? The answer is here. 99.9% in sewage is water molecule. So water is, the wastewater is very important water resources for us, especially for those areas have shortage of water supply. And later on, people also look at the wastewater in detail and found that some pollutants in the water, like nitrogen, phosphorus, these kind of elements in the water, in the sewage, they are pollutants. They can cause problems like eutrophication, the red tide, the algal bloom, but also they are nutrients for the agriculture, for our human being, food supply. So to recycle recover the nitrogen and the phosphorus from sewage is another research topic. So it is also flow of resources. And also sewage or wastewater is also flow of energy. You see, where is the energy? The sewage contains organic matters. The organic matter has chemical energy. If we can release it out in some form, like methane, biogas, under anaerobic condition, then we can recover this part of the energy in the water. Of course, the energy in the water not limited to the organic matters, but also to heat or some other forms. And now, people also realize that wastewater or sewage is also flow of information, especially demonstrated in the COVID period. The worldwide application of wastewater-based epidemiology tells us that we can extract information from the sewage, from the wastewater, by using some molecular method, by using some chemical method. Actually, now, if we can get enough information from the sewage, we can get the other informations beyond the health part. For example, the illegal drugs. For example, the smokers in the high school. One of my postdocs in the group now just proposed a proposal to use the sewage-based surveillance to look at the smokers in our community. And our research focus is on sewage. But which part? We are not chemists. We are microbiologists. So our focus in the sewage is the microbiome. So what is the microbiome? You heard about the microorganisms. But what is the microbiome? This is a new term just invented some years ago. That means all the genetic materials of all microorganisms in an environment, like the reactors, or our beach water, or the soil, all this kind of environment. If you extract the DNA and sequence the genetic materials of all the microorganisms in the format of ATGC, then you can know what are they and who are they, what they are doing, why they do that, and more importantly, how and when they do that, so we can engineering that. We can design and operate our reactor in a better way. That's our belief. And the microbiome, if you look at the definition from nature, that's all the genetic materials within a microbiota. The microbiota is an entire collection of all the microorganisms in a specific niche, specific environment, okay, or such as the human gut. And about 9 years ago, in 2016, US government, White House, Obama period, they announced this National Microbiome Initiative. That because microbiome had impact on all the aspects which I put on this picture. For the environment part, we have air microbiome, which may affect our immune system, affect our skin microbiome. And for the water, they may cause a hygiene problem and also microbial quality problem. For the soil, they may affect the uptaking of nutrients of the crop. And for the human system, they also have very significant impact. Let's take the disease as example. Previously, we know human has different disease, but the recent findings, scientific research found that many of these kind of diseases had a relationship with the gut microbiota, with the gut microbiome in our stomach. So previously, we guess there could be some relationship, could be some association, but we cannot confirm that. But now with the microbiome study, we know there's a strong link between some kind of like a cancer with the microbiota in our human gut. And also, if we can make good use of a microbiome, then we can have this kind of so-called bioeconomy to generate the bioenergy and the bioproduct and recover the oil from the food. So microbiome had impact on all these aspects. If we look at our study on microbiome or microbial ecology talking about the interaction of different species in an environment. So basically, it's talking about the microbiota or microbiome. And I will put them into the research method and our methodologies into 3 different stages or 3 different versions. Yeah. In the version 1.0, we mainly use the microscope and isolation to study the microorganisms. Under microscope, we can observe the shape of different bacteria or fungi and to identify, okay, what kind of species they are. But it's very limited because they all look similar. And if you look at this picture, the photo that's taken by Dr. Chun Xiaowang, one of my former PhD students, you look at it, they're all similar. But there could be 100 different species there, have different functions. And we also can get some colony on the petri dish by providing the culture media. They form single colonies, then we can start from there to study what kind of function they may have. But many bacteria, they cannot grow on the petri dish. So due to this kind of limitation of microscope and isolation, although we studied some species of microorganism, bacteria, we accumulated some knowledge about the physiology, metabolism, the K in them. So this is a solid knowledge about microorganisms, but still very limited portion had been studied, had been discovered. There are a lot of dark matters in the nature which not known to us. It's unknown microorganisms. Then you may ask, how you know they're unknown there? Then that's a logical question, right? How you know they're unknown there? Yes. If we're just based on the isolation, just based on the microscope, we can know. We cannot know. We cannot know how much unknown is there. But using the molecular method, using DNA sequencing, we may know there are significant portion in the nature, in our gut, in our reactor, in our beach water or soil, not isolated yet, not being observed on this microscope yet. And so in the version 2, using some marker genes, we can use those marker genes as a fingerprint to identify some of the microorganisms and give a long list which beyond the limited list based on the culture, based on the microscope. But this only provides a name list. We still don't know what kind of function it may have because we only have very small fragment, fingerprint fragment can tell us who they are, but we don't know what kind of function they may have. And in the version 3, when we have the next generation sequencing, when we can get huge amount of DNA sequence with a very cheap price, then we may sequence almost all the organisms in an environment after DNA extraction. As long as you can extract the DNA out, it can be sequenced. And with this kind of sequence of microorganisms, we can put them together by overlapping by some other algorithm and make it longer and get the so-called draft genome of bacteria. And here is an example demonstrates this kind of revolutionary finding of microbial dark matters, which never been found before. And this paper published on Nature Microbiology about 8 years ago. It found 8,000 new bacterial genomes, which never been reported before. So if you look at a Nature review paper about DNA sequencing, it said that in long view of history, the impact of DNA sequencing will be on a par with that of microscope. So basically, we can simply say sequencing is the next microscope for microbiology. Then let's look at the sequencing. What happened during the past more than 20 years? What is the price of the sequencing? And here, there are two curves. The first curve is Moore's law. I think most of people are familiar with this one. It's talking about the semiconductor price getting half after every 2 years. Yeah. And the green one is Carlson curve. It's talking about the sequencing price starting from 2001 to 2021, about 20 years. As you can see, back to 2001, for one human genome, it will cost you $100 million US. 100 million US dollar. But by 2021, it's already below 1,000 US dollar for one human genome. You may ask me, what is the current price? Okay. Look at this Nature technical feature articles. In the year of 2023, about 2 years ago, they summarized the development of the so-called short-read sequencing method and said that for one human genome, if you sequence it using Illumina, the US sequencing technology, it will cost you $200 US in the year of 2023. But if you sequence it using China sequencing technology, MGI, then it's only $100 US. And also mentioned by this article, they said that there is an old saying in the field of technology: nobody ever got fired for buying IBM. A reference to companies once ubiquitous computers. Replace IBM with Illumina, the US sequencing technology, the sequencing company, a biotechnology company in San Diego. The same could be said for DNA sequencing today. That's from this review paper. But it could be wrong. If you buy Illumina for a Chinese company now, it could be fired. Why? Because Illumina had been put into the unreliable entity list by Chinese government back to February 4. Here, yeah, I think February 4. So it's just after the Lunar New Year. And one day after that, if you look at the stock market, MGI, at that day it's about 45 something, and shortly doubled. And even now, yesterday, it is 75. So pay attention to this kind of development. It will not only help you to publish a paper on scientific journals and also can earn some money from stock market. Okay, almost the same time, even 1 month earlier than that feature article, the Nature Measured published articles about the measured of the year. They are talking about the long-read sequencing. The previous one is talking about the short range. Short read means that you only can get a few hundred base pair. But for one bacterial genome, it's 1 million base pair. For one human genome, it's 3 billion base pair. But the long-read sequencing can be much longer than short range. Depends on which platform you are talking about. No, sorry. There are PikeBio and Nanopore. Nanopore is the platform we are largely using now in my lab. And PikeBio is also American-based company. Nanopore is UK-based. And now MGI, they also have this long-read sequencing technology just announced last September. So lung rates can easily give you a few thousand, and even up more than 10K, 50K. The longest sequence, just one physical fragment, could be a few million base pair, and almost one bacterial genome. So long range is also very important development in the sequencing field. So after we look at it, then what is next? What is next? What is the next version? We talk about the version 1, version 2, and version 3, 1.0, 2.0, and 3.0. For 1.0, we only have very limited knowledge. For 2.0, we get a list about the bacteria in different environments. And for 3.0, we get the genome, we know the function. But what is the next? The next one, if we learn from other fields, like the environmental analytical chemistry, it's talking about the distribution and concentration of chemical pollutants. If we learn from that, they are very similar. I think for the next version, microbial ecology 4.0, it should be environmental analytical microbiology. We will analyze the microbial cells and the genetic elements similar to the chemical pollutants, very similar to the chemical pollutants. So we can borrow the idea, the principle, the protocol from the environmental analytical chemistry, and to overcome the challenge when we do the analysis without this analytical aspect. For the current challenge, we have the cross-condition, the sample study comparison, and we also may not have one complete observation of the dynamics. We only observe partial dynamics. And also, we may have some misleading correlation if we're based on the non-quantitative information. And now, the urgent need is absolute quantification. With the absolute quantification, we can get meaningful biological insight and also some comparative analysis about the different samples. So let's look at the quantity, the amount of the bacteria, microorganisms in different environments. Here is the figure summarized by Dr. Wang about the bacteria concentration in different environments. For feces, you can see it's a very large range, 10 to the power of 9, or sometimes even lower to 10 to the power of 8, then up to 10 to the power of almost 13. So very wide range. So if we only use the relative abundance, relative amount, it cannot tell exactly what happened. The same for the water, for the soil. You also see very wide range of the absolute quantity. So we definitely need the absolute quantity information to look at the distribution of the microbial pollutants and especially the pathogens in our environment. The question is how? How you can get the absolute quantity? If we look at what is the practice in the environmental analytical chemistry, we will know the answer. For the environmental analytical chemistry, they add some compounds with a known amount. And here, same, we can add the internal standard of some DNA or cell with known amount to a given volume of the sample. Then you know that is the benchmark. And when you sequence them, then you can get the amount of the internal standard in the total sequencing data set. And based on that, you can convert it to the absolute amount of all other different species as long as you can sequence it. But for when we select the internal standard, there are two different types of internal standard. One is starting from naked DNA, and another is from the cell. The difference between the two are very clear. When you spike, when you add the naked DNA, you didn't simulate the cell breaking process. But when you add the cell into it directly, you will also simulate the cell breaking process. So that will be more close to the reality, to what happened in the DNA extraction process. And another question we need to answer to evaluate is how much we added in. For the internal standard, if you add too much, then most of the sequencing result will be on the internal standard, not on your target population. But if you add too low, then you may have the biased quantitative result. So these two things must be balanced case by case. For different type of environment samples, we should do the evaluation to see which dose level could be the best and should be optimized. And here is just an example, show you how we do that. This is done by another PhD student. This student used this labeled E. coli spike in and add into the beach water, into 1 liter beach water, and with a known amount, and extract this spike in, this internal standard, together with all other bacteria in the beach water and into the DNA, and sequence it. And when we get the data set, we will know how much of this E. coli in the sequencing data set. And based on that, we can establish a scaling factor. You can take this scaling factor as a kind of recovery efficiency. And based on the scaling factor, we may calculate back for absolute amount of all other different bacterial species, including the pathogen and antibiotic-resistant genes. So using that, we can get the absolute amount of bacteria in the unit of cell per liter or cell per milliliter. And with this kind of information, what can we do? How it can help us in the application of engineering. First of all, we may get the quantitative microbiome profiling. So you can get the profile of the different species in an environment. And second, you can do the quantitative microbial risk assessment. And based on this quantitative information, we may do the modeling work and to achieve patient control. So that's the whole purpose for us to do this kind of absolute quantification of microorganisms. Let me give you 2 examples here. The first one is about the dynamic of microorganisms in anaerobic digester, in anaerobic digestion reactor. And here is the full-scale anaerobic digester in wastewater treatment plant. And anaerobic digestion is the process which we used widely all over the world, also including Hong Kong, to treat our wastewater, and convert the energy into biogas form and recover the energy. So, and also using this process, we may recycle the nutrient and kill pathogens. But in quite long time, we don't know what are the major populations, what is the absolute amount, concentration of different species in a reactor. And in this study conducted by Dr. Wang, first of all, we get the genome of all the microorganisms in this reactor. And then, based on the absolute quantification result, we may calculate the growth rate and the decay rate constant of different bacteria. So we may calculate the doubling time for different species and the reduction time to achieve 90% reduction, how much time it will be needed. And based on that, we can design the solid return time and decide how much volume, how much land will be the reactor to design the reactor in a better way according to different requirements. And the second example is about the microbial risk assessment and the water quality index. We know that when you go to the beach to swim, you may need to look at the report of the E. coli count currently. which measured by EPD every day. They use the ecology, one of the fecal indicator, to indicate the water quality. But that's only part of the microorganism, the pathogen in the beach water. It's not that comprehensive. We say why they do that instead of measure all, because the methodology is very limited before. But now, with the metagenomic sequencing, when you can sequence all the different bacteria, all different pathogens, and then we can get a more comprehensive water quality index. And for this aspect, first of all, we want to answer which microbial pollutants are there and what are the quantity of the concentration. And based on that, we established sub-index on different aspects, on 4 different aspects. First of all, still the conventional fecal indicators included. And we also extended it to other pathogens which are not covered by the current water quality index, and also including the resistome and the mobilism. All these kind of things can put together as an overall risk index to represent the comprehensive assessment result of the microbial risk assessment level. So this risk assessment framework can prioritize the microbial risk for the informed public health action. And one of the aspects which I talked in the second example about microbial risk is the resistome. So what is the resistome? That is also a new term for many of you. Resistome means the collection of all the resistance genes in an environment. It's about the antimicrobial resistance. The antimicrobial resistance or antibiotic resistance is an emerging public health topic under the One Health concept. Under One Health concept, we consider the health of the human, health of animal, and health of the environment all connected. The antibiotic resistance is one of the good examples for One Health concept. If you look at the antibiotic use in different sectors, 30% used for human being, 70% used for the animal. And after being used, 80% of antibiotic used by human or animals will go to the environment and become the pollutants in the environment, together with the resistant bacteria and the resistance genes. And all these are considered as pollutants. And this topic has drawn a lot of attention, not only from WHO, but also from UNEP, United Nations Environment Programming. UNEP published a report the year before last year, about 2 years ago. It's Bracing for Superbug, Strengthening the Environmental Actions in the One Health Response to Antimicrobial Resistance. And I'm one of the reviewer for this report and contribute to the opinions and scientific evidence. If you read this report, you will get this key message. This report provides evidence about the environment play a key role in the development of AMR and the transmission and spread of AMR. And the prevention is at the core of the action, and environment is a key part of the solution. So that's a key message from this report. And my research group has been working on this topic for more than 15 years, And before the pandemic, in the year of 2019, I organized an international conference on this topic, environmental dimension of antibiotic resistance. And about 350 people attended the conference, including George Fugao. At that time, he was the director of China CDC, and also Professor Martin Plater from US, and Sally Davis from UK. And our group developed a metagenomic analysis tool for people to use the metagenomic data set to analyze the resistant genes in different environments. And this tool has been visited by the researchers worldwide during the past 9 years. Yeah. And also, we work together with the global research community, the different co-authors from 19 countries, 30 institutions, totally about 40 co-authors. We published a paper and proposed that we should use the universal unit for quantification of antimicrobial resistant genes in the environment sample. The unit is copy per cell. And using the methodology of metagenomics, we can have a very comprehensive profile of resistant genes in an environment. From this figure, you may see these are the different types of environment sample, and these are the resistant genes belong to different classes. And here, just to shows if we sum them all together for different environment, what could be the level of the resistance genes as the pollutants. In the marine water, which considered as clean, not polluted yet, the level is very low. But when you look at the chicken feces or swine feces, or even sewage from a human being, the level is already quite high. It's about 2 magnitudes higher than the natural level. And using metagenomic sequencing, you can sequence thousands of different resistant genes. The question is that, are all of them important? How we can pick those important ones, prioritize our action. We did some research on the high-risk antibiotic-resistant genes, and based on 3 different criteria. First of all, whether these kind of resistant genes be enriched by human activity, and whether they're carried by the mobile genetic element can be transferred from one pathogen to another, and whether they has been hosted by the pathogen. So based on these 3 criteria, starting from 4,000 different resistant genes, we have a short list about 120. So these are the resistant genes we should put our focus on. We should prioritize our action to have more intensive monitoring and control strategy. And also, very important in environment analysis, we should know what kind of species is a carrier of the resistant genes. If the resistant genes is carried by pathogen, the risk level is higher. But if carried by some natural native bacterial species, the risk level is not that high. So how can we assign the resistant genes to a specific species? Species level resolved profiling of the resistant genes. We can use the long-range sequencing. As I mentioned before, long-range can give us easily about 10K or 50K. Then it can give us more information about what kind of species this DNA sequence represent. So this is another paper we published this year by a PhD student. It's also sitting in the floor. We use the long-range sequencing to identify the resistant genes at the species level. So I can identify whether these are the pathogen or non-pathogens. And also, using metagenomic sequencing, different from the culture-based. For the culture-based, when you give them petri dish, when bacteria can grow, they're alive, they are viable. But when they cannot grow, they may not be dead, okay? They may not be dead, they just cannot grow on it. But for the metagenomic sequencing, whether you can differentiate the live bacteria and dead bacteria, the answer is yes. This is the method we published using the PMA, which special treatment of the environment sample can differentiate the live and dead bacteria by metagenomic sequencing. So this gives us a more powerful tool to differentiate the live and dead bacteria in the environment. Because in the environment, both live and dead bacteria are there, not like in the clinical field. And most of them, they are live bacteria. So for the environment samples, if we cannot differentiate live and dead, then we may not assess the risk exactly. But using this method, we can to differentiate the viable bacteria and the dead bacteria. And for technical detail, I'll escape it. If you have interest, read our publication. And using the established method, we are working on a sim-based research project. This project had lasted for 5 years. This year is last year. And for this project, we work together with colleagues from biological science, from chemistry, from environmental chemistry, from human microbiome, from the environment engineering, and also from computer science, from public health. Work with colleagues from different disciplines together to work on the antimicrobial antibiotic resistome flow from the pollution hotspot to the environment and explore the control strategy. The whole idea or whole ultimate goal is to mitigate the burden of antibiotic resistance and Hong Kong citizens are exposed to. We will provide the systematic surveillance result and give a local resistome map plus the risk assessment and some control strategies proposed. And lastly, I will use a few slides to share with you some ideas. First of all, I think it's time for us to change our mindset. Now, One Health is a very popular concept. And yesterday in school public health, they have the first lecture on this topic about One Health. And there's a few journals with One Health as the name. But for me, One Health is not that new concept. As a water engineer, and when we study the water treatment, the wastewater treatment, I think that's already a very good demonstration about One Health. One Health talking about animal, environment, and human health all connected. You cannot solve the problem just within one sector without your effort in other sectors. For me, I think it's very natural historical lesson from the water treatment. Back to 100 years ago, many people, they drink the untreated water, they get sick. Some even be killed, died. But after the water been treated using chlorine, then this problem had been largely solved. And later on, when the wastewater also be treated and put them together, the water treatment, including the drinking water treatment and wastewater treatment, save millions of lives. That is a very good demonstration about One Health concept. So for me, I think for many water engineers, wastewater engineers, One Health concept is not new. That's something people take it for granted. Take it for granted, like drinking water. People just think that drinking water must be clean. But actually, back to 100 years ago, it may not be that case. And also, another important thing I think we also learn from engineering practice. If we do not spend money at the source control, we'll spend more money in a hospital, in a community, and in an animal farm. Because the problem will be diluted and getting bigger and not easy to be solved. And usually, the solution at upstream is much cheaper than the solution at downstream from engineering point of view. But how to convince people to do that, it's very difficult. Not that easy to convince people to take action earlier for prevention. And here, I also would like to share with you a Chinese classical stories. First of all, it's a fake story. It's not faked by AI. It's faked by human intelligence, okay? Not fake story created by AI, but it's a fake story created by HI, human intelligence. Fake story, not true. But the lesson is real. It talks about Bianque and his brothers. Bianque is a very famous surgical doctor in ancient China. And one day, the king asked, who is the famous? Who is the best? Your three brothers are all skilled in medicine. Which of you is the best? And Bianque answered, my first brother is the best, and the next is my second brother, and the last is me. The king is very surprised and further asked that, why? And Bianque replied, with all this, I think many of you already know that, so no need for me to repeat it. And also the Chinese original version is here. This is just some English translation, may not be that correct. And the lesson from this fake story is that prevention is better than control, which in turn is better than remedy. After the event, tackle the AMR, the antimicrobial resistance problem, in the environment before it comes back to human and animal is important. I think that's some lessons we may learn from it. And also now, I think in addition to the fundamental research, and we should pay much more attention to translational research. How can we transfer our research into some real impact? I think our research on the microbiome, sewage microbiome, environmental analytical microbiology has demonstrated its power in the pandemic period of Hong Kong. And when we use the sewage surveillance to identify those hotspots which for the following public health action. And this has been demonstrated already significant, demonstrated clearly, and got some international award and also the local government award for myself and also for the whole team, and also get HKAE Grand Award in 2023. And also, not only for the social impact, we may also can transfer our research into some commercial product and get some money from it. This is some recent news about from our partner, MGI. They put our invention into a package, for the monitoring of sewage, including this auto-sampler and also including our analysis pipeline into a whole package for the real applications as a commercial product. And hopefully, working together with the world-leading high-tech company, we can upgrade so-called made in China to invented in China. And here is just a few very short take-home message. First of all, environmental analytical microbiology is a frontier in environmental engineering. Absolute quantification using metagenomic sequencing is one of the cornerstones of environmental analytical microbiology. Environmental analytical microbiology, of course, is much more beyond the metagenomic sequencing for absolute quantification. It's much more beyond that. And development of environmental analytical microbiology will reshape our design of wastewater treatment reactor and also improve our microbial water quality assessment method and provide a new tool and commercial product for engineering applications in the future. Lastly, I will thank the hardworking team and the Sim-based research fund of the Hong Kong RTC, Hong Kong government. They provide a lot of help on the sampling and the applications, and also the high-performance computing facilities of Hong Kong U. I see Mr. Guang is over there. Thank you for all the help all the way. Okay, thank you. Thank you for listening.